Right. Well, we're here with, I should say, Professor Mike Lustgarten. I can get away with calling him Mike because, well, we know each other quite well.
Yeah.
Hi, Mike. How are
Hey,
you?
Steve. Good.
So we've been having a bit of a chat today about the new paper, the new paper that shows the link between the gut microbiome and Alzheimer's. I mean, it's something that's been a fairly common topic in I would say, the last year or two. And there's always been the suggestion that there has been a link. And I think really this paper just, for me, it really built on that and said, look, clearly some forms, if not all forms of Alzheimer's, definitely have this bacterial link. But obviously, you found it as exciting as me, didn't you?
Yeah. So I think we should. So the first and easiest, low-hanging fruit to get after with that is the LPS. So increased LPS in the blood was associated or significantly correlated with a higher amyloid burden in the brain, right, in people. So
Right.
LPS, as you know, as most people know or may not, it's a bacterial metabolite. It's only found in the bacterial outer membrane of some bacteria, not all bacteria.
Mhm.
And if it's in the blood, there's a decreased gut barrier function, increased intestinal permeability. So right off the bat, the most definitive data shows a role for, or suggests a role for, intestinal permeability and decreased gut barrier function on Alzheimer's disease or amyloid burden. The more interesting data is the acetate and the butyrate correlations, because superficially it seems to be counterintuitive. So I think that's where the story gets interesting. I can keep going there, but I don't know if you want to interject before I go to 5 or 10 minutes.
He's not joking, guys.
Yeah.
He really could. But not that he isn't a fountain of knowledge where the microbiome and the interactions with it are concerned. So he's definitely on the ball. But yeah, it is an interesting thing with those two short chain fatty acids. We know from previous rodent studies, which I've mentioned in the article, that butyrate or butyrate, as some people call it, butyrate, I call it, that does seem to have beneficial effects in terms of health and even longevity in rodents. And there's even some tantalizing data for its effects in humans. It was great to actually see in this study that built upon that and sort of further reinforced that butyrate is definitely beneficial. We're still a little bit question mark on the supplementation of it, but certainly supplementing butyrate in rodents, and obviously mice are not men, but it did seem to improve their health and longevity as well. So I don't think it's a stretch thinking that even supplementation might help. But of course, if you can get it in your diet, butyrate, even better. And obviously, there's all kinds of foods that have got butyrate in them. But I'm sure off the top of your head, you could probably tell us which ones are the best or better.
So just going through the butyrate story. So first, it's a gut bacterial derived metabolite. So if you take animals that don't have microbiome, germ-free mice, they're grown in a bubble, and you do fecal metabolite analysis, there's no butyrate in their feces. In comparison with the conventionally raised animals that have microbiome, when you look at their fecal metabolite levels, they have butyrate. So butyrate is exclusively produced by the gut microbiome. So that higher levels of butyrate in the blood were correlated with a lower amyloid burden. That strongly suggests a role for the gut microbiome on impacting Alzheimer's disease. Now, in terms of getting it from food, so only foods like butter. I think butter is actually the food that's got the highest concentration. But granted, it's not a very high concentration of butyrate in butter. But the best way to do it is by increasing soluble fiber intake. And this is mostly by eating lots of vegetables, because vegetables per calorie compared with all the other food groups, they're not very calorie dense, but yet they've got a high concentration of this soluble fiber, which the gut bacteria then convert into the short chain fatty acids, acetate, propionate, and butyrate, not just butyrate. So, but just along those lines too. So of those three short chain fatty acids, acetate, propionate, and butyrate, Butyrate is actually found in the lowest amounts, both being produced by the gut microbiome and in blood. So the ratios, it's generally, 3-to-1 acetate to butyrate in terms of its levels in feces and in blood. So butyrate is a very small component of the fecal metabolites that are produced. So, but nonetheless, so then in terms of supplementing with butyrate or supplementing with short chain fatty acids, this has been done in animal studies with some beneficial effects on muscle. That may not be the most optimal approach. And what I mean by that is, so when you eat it, it's going to get absorbed into the through the small intestine into the blood. So that's a good thing. But when considering that fiber is converted by the gut bacteria, which are mostly heavily concentrated in the colon, which is downstream from the small intestine. So fiber goes to the colon, and then that fiber in the colon is changed into the short chain fatty acids, where they then act on the colonic epithelial cells and then diffuse into the blood, where you see them systemically as they were in this paper. But by eating them, are you getting the full effect if you bypass the colon? Do you need the colon for the health related effects of some of the short chain fatty acids? That it hasn't been studied properly. So I'd argue that actually getting them through fiber is likely going to be the most optimal approach. But again, there are animal studies showing that just eating, supplementing with them, or in the case of sodium butyrate, can have health benefits. But again, maximal versus some benefits, that's the case. So the interesting thing here then is not so much, so the butyrate story goes along those lines of the gut microbiome affecting, Alzheimer's disease risk, but then it's acetate, which then is the debatable issue in this paper, which is interesting for its interpretation.
Yeah, that was a bit strange, really. The high acetate levels also correspond to the presence or the increased presence of plaques, or this particular amyloid plaque associated with Alzheimer's. It's very interesting. Not quite sure what to make of that, really, as it seems to be contradictory. I mean, have you got any initial thoughts on that?
Yeah, so I think, we've got to see what the data is and then build a hypothesis after that. So that's what I feel like I've done. So there's a couple of steps in that process. So one, you've got the high butyrate levels. So for there to be high butyrate, you'd have to have a high butyrate associated or correlated with lower amyloid plaque, right? So for that to be the case, you'd have to have high acetate, right? Because actually how butyrate is made, one of the major pathways that butyrate is made in the colon is by converting 2-acetate. So it's a 2-carbon fatty acid, which is converted into the 4-carbon fatty acid butyrate. So that's the predominant way that butyrate is formed in the colon, by having a large amount of acetate converted into butyrate. So then you'd predict that the high acetate should be correlated with the lower burden of the amyloid plaques, right? So that actually would argue against, but that's not what the data was, right? It was actually the opposite. So low acetate, and then there was high butyrate. So that would suggest that in the people who had low amyloid plaques in the brains, they may have an increase in the acetate consuming bacteria to make the butyrate. In that case, the circulating levels of acetate would expect it to be lower in the subjects who had lower amyloid burden, which is what you saw, right? So that's the first interpretation of how I would see that data. Now, conversely, for the people who had a high amyloid plaque burden, who had high acetate but low butyrate, right? So then why would they have high acetate? So acetate isn't exclusively a gut microbiome produced metabolite. So it's also produced by our own systemic physiology. So when you have glycolysis, glycolysis will, the end product of glycolysis is pyruvate, and then pyruvate can get converted into lactate or acetate. So mitochondrial dysfunction is a well-documented feature of Alzheimer's disease. And when you have mitochondrial dysfunction, you're going to have an increased reliance on glycolysis systemically all over the body. So in that case, I'd argue that for the person who's got Alzheimer's disease, mitochondrial dysfunction, increased reliance on glycolysis, increased pyruvate, because pyruvate can be converted into acetate. That would explain why they would have high circulating levels of acetate, but low gut microbiome levels production of acetate and correspondingly low gut microbiome production of butyrate. So to me, that's what would make the most sense. Unfortunately, they didn't measure anything related to mitochondrial function, and they even speculated on the gut microbiome dysbiosis, but they didn't actually measure, the composition of the microbiome to actually show this microbiosis. So all we can do is, make reasonable guesses based on the data.
Well, that's what scientists do. But I think as hypotheses go, it's pretty good. It's a good start. And of course, mitochondrial dysfunction is such a big element of aging, or proposed to be such a big element of aging, that it appears not once but twice in the hallmarks of aging. First as a primary reason, but it also gets another mention as a tertiary reason as well. The actual dysfunction itself, and the breakdown of energy production. So that's interesting, where that acetate could be coming from.
Yeah.
So I mean, I thought it was a really solid paper, really interesting, and it's could it be another brick in the wall for your proposal that, you know what I'm going to say? Could it be that the gut microbiome is another potential hallmark of aging?
Yeah, and actually, you could make the argument too, as I did in the book, not to plug the book, but that the microbiome actually plays a role in all of the hallmarks of aging. So whether you include the microbiome as its own hallmark, or add, include the data that shows the microbiome contributing to all the hallmarks of aging, I think that's an important thing. Actually, I wrote a paper on, so when the classifying aging as a disease. So Sven wrote a paper about it, and then somebody else wrote a paper about it. And I wrote a paper a few years ago saying, classifying aging as a disease, don't forget about the microbes, because microbes have been involved in all these, aging related diseases and processes. So yeah, for sure. I think the microbiome definitely contributes to the hallmarks of aging. But in terms of the microbiome Alzheimer's disease risk link, when considering that butyrate, like I said, is a gut microbiome metabolite, gut microbiome derived metabolite, whether that's supplementing with butyrate to get it into your blood, or increasing your fiber intake. I think these are strategies that, randomized controlled trials should be going after. And as far as I know, that's not the case yet, but hopefully soon.
And I think if memory serves, butyrate is actually fairly inexpensive as a supplement itself. I don't think it's particularly costly, so it's not like some of the more expensive popular supplements at the moment, things like NR and NMN. I have to say that slowly, otherwise it comes out funny. But I'm very interested in those as well, but they do have a very high price point, obviously due to the production methods that are required to stabilize them, etc., which does make them quite costly. So butyrate, maybe a low-hanging fruit here, maybe. And it also suggests that correcting dysfunctional mitochondria, a la SENS Research Foundation and their MitoSENS program, could also be very promising in that direction too, as well. So,
yeah.
But as you say, all the hallmarks are definitely linked. Inflammation is definitely, as Aubrey has said at one point, inflammation is the sort of thing that binds them all together, almost like a communication network between all the different hallmarks. But absolutely nothing works in isolation, and the hallmarks are certainly not going to be an exception. But yeah, aging is a disease. Slightly off topic, but I can't help but say that there's a lot of argument whether it is or isn't. Some people say that calling it a disease stigmatizes it. The FDA and EMA are certainly not keen on the idea, shall we say. And obviously, from the point of view of endpoints of clinical trials, it's not really practical in that sort of sense. Just say aging, that's not an endpoint. But I have said it before, but I have suggested that aging isn't necessarily a disease anyway, but by its very definition, it could be potentially classed as a syndrome, because it is a collection of symptoms often occurring together, and that in a medical sense, I would suggest that aging is a syndrome. But there you
Yeah,
go.
that's a fair point. I don't
That
know if I'm not, I don't know if I'm so married to the ethics of aging as a disease or not, right? I just want to see it conquered.
yeah, it's a complicated issue,
Yeah.
and the important thing is I see the point of view of both sides. I can understand why mechanistically some people might think it is, and why some people might think it isn't. But importantly, from a regulatory and quantification point of view, it's important that we reach some kind of consensus here, which is again why I think biomarkers, we're always going on about biomarkers, are so important. And this paper here actually gives us some
Yeah.
really, really good biomarkers, especially for Alzheimer's. But yeah, although I will say that there's increased evidence now that there's more than one kind of Alzheimer's disease, because there do appear to be many different sort of neurodegenerative conditions that were once lumped in as, oh, it's Alzheimer's, it's Parkinson's, but it seems to be that it's more nuanced. And whilst there might be, in this case, there may be some people with a bacteria driven Alzheimer's, it may not be the case in with other people, which is why they found it so hard to nail down.
Yeah.
Any thoughts on that? I mean, could we be? Do you think we might be looking at multiple types of Alzheimer's, each with their own individual sort of nuances?
So unfortunately, I haven't seen data that compares, bacterially driven Alzheimer's versus virally driven versus fungi driven. But yet
Mm,
there is data that each of those microbes are found in the brains at a increased incidence compared to control subjects who don't have Alzheimer's. So I wouldn't be surprised to see, the signature, whether it's inflammatory, metabolites,
mm.
pick your biomarker of the signature for a bacterially driven Alzheimer's disease. It wouldn't surprise me to see that signature would be different from a virally driven or a fungi driven. Now, there may be some overlapping mechanisms for each of those, different microbes driving the pathology, but I wouldn't be surprised to see independent variables that are different with each. But still, even the idea that Alzheimer's has a microbial component, how much of it the microbial component is driving the disease, or it's just the correlation of the disease. I'd say that that's still not the majority opinion in the field, unfortunately, from my perspective. And we'll find out eventually in the future, right? How much microbes are actually driving the disease? Is it all of it, or is it the combination of poor diet and pick certain genes, whether it's ApoE or other genes, but nailing down all the variables that can, lead to Alzheimer's disease. I mean, it's obviously important, right? So
It is, and I think this paper is a big step forward to that end, and as well as the biomarkers angle as well. I think they could be pretty superb, and I understand not that difficult to actually to measure, because it's just in the blood. So I understand that it wouldn't be too difficult to measure things like the presence of short chain fatty acids in blood. It's
So the short chains in fatty acids are actually difficult. So
hmm,
they get metabolized very quickly. So you'd have to have either very high or well, in theory, very high levels of it. So and I say that they're difficult because the core at Tufts tells me all the time how difficult it is to measure them in blood.
hmm.
Now in stool too, it's relatively similar. Even though many studies have reported the fecal levels of the short chain fatty acids, they're also metabolized very quickly. So making sure you get the stool sample immediately and then putting it in storage solution immediately, as opposed to just freezing the stool sample and hoping that it's not, the short chains aren't degraded. But yeah, I've got one of the grants I have in submission right now. I have a plausible, what I think is a plausible hypothesis for how the short chains are basically at the heart of all things microbial in terms of health-related effects. So real quick, the short chains are fatty acids, which means they are acids. The gut in the colon, to have a good microbiome, the pH has to be lower, not higher, which is more towards acidic. So when you have a higher concentration of the short chain fatty acids in the colon, that acidifies the gut, or mildly acidifies the gut, where it increases the growth of, the quote unquote good bacteria and minimizes the growth, as we talked about this a little bit, minimizes the growth of some pathobionts like enterobacteria, which can produce metabolites that have been shown to negatively affect kidney function, can negatively affect muscle. So if you don't have the short chains, the pH of the gut will be higher. You'll have more of these quote unquote bad bacteria, which produce more of these bad metabolites, and that's bad for systemic physiology. So I think that the short chain fatty acids are going to be shown. And, this goes beyond all of the studies, or most of the studies that are out there, that just look into, what are the differences in microbiomes, microbiome between cases and controls, whether it's Alzheimer's disease or pick your illness of choice, right? So besides that, how's it happening? How are the different microbiomes contributing to these phenotypes? And the short chains, I think, are essential in that process.
And we haven't even mentioned the other short chain fatty acid as well that was elevated with people with Alzheimer's, and that was valerate.
Right.
So any thoughts on that one?
So valerate is also, when compared with the acetate, propionate, and butyrate, valerate, so that's a 5-carbon fatty acid. And then also, the short chains go up to 6 and 7. And then we start to get into the medium chains with 8 carbons. But so valerate at C5, it's in very small amounts. It's at least when it's in the gut. So I imagine they are comparing smaller amounts with slightly less small amounts. But nonetheless, from my experience in reading in the literature, valerate is not necessarily produced in high amounts by fiber fermentation, but more of protein fermentation. So, and I think in this case, higher valerate was associated with or correlated with the higher amyloid burden, which would make sense in terms of, so the higher levels of circulating amino acids have been linked, for example, to obesity. So I bet that that's got something to do with this story, so more proteins and more amino acids in the blood, more amino acid fermentation in the gut, more production of the amino acid fermentation metabolites like valerate. But again, this is I hate to say I focus almost completely on the acetate, propionate, and butyrate story, but that's like maybe 95+% of the quote unquote short chain fatty acids that are produced in the gut. I tend not to focus so much on the short chains like valerate. But what's interesting though is there are branch chain fatty acids, although they didn't measure it in this story, which are also protein fermentation by the gut bacteria metabolites. And there are many studies showing that fermenting dietary protein into these branch chain fatty acids, branch short chain fatty acids, is bad for health. So I wish they actually would have measured some of those.
Yeah, and obviously the search is now on. The researchers' next step is to track down these individual bacterial populations and see if they can identify the particular culprits. And that for me is also quite interesting because there's good support for what you do of dietary interventions and modifying your lifestyle, and maybe not eating a bacon sandwich every morning, and trying to sort of include more sort of fiber especially, and vegetables in your meal. Not necessarily go vegan. We're not suggesting that. And there is obviously, as you often say, there's no perfect diet. You should eat a diet suited to your microbiome
In the biomarkers.
And the biomarkers, yeah.
Yeah.
So everybody is different, and so is your microbiome. But from a sort of a prevention point of view, as well as doing those things, this study really and tracking down these bacteria, these particular subtypes of bacteria that are guilty of being causing the problem, that also opens the doors for a potential therapy, right?
So yes and no. So at first, I want to say I'm skeptical that so let's say they identified there are many bacteria that are known to produce the short chain fatty acids, including butyrate. So if it was as simple as saying, okay, we know these bacteria produce butyrate. Butyrate is associated with less, amyloid burden. Let's just give you this bacteria. So if you took that as a probiotic, okay, now you need the prebiotic for those bacteria to grow. For whatever reason, the people who had an increased amyloid burden don't have the substrate for those bacteria to exist. So if you're going to give that bacteria as a probiotic, you've also got to give what they like to eat, which then again goes back to you've got to have lots of soluble fiber for those bacteria to use that to continue to live in the colon. So you can feed them continuously, if you give the probiotic, that's the seed. And if they don't have the prebiotic, the fiber that they eat, right? So let's say you don't have the prebiotic that those bacteria that you're giving like. You're just likely gonna, poop them out because your body doesn't need them, or they're just gonna, die off because there isn't anything sustaining their growth. So where was I gonna go with that? Okay, so then that's one side of the story. The other side of the story then is, which is amazing that I'm even on this side, but fecal transplantation. So instead of just going after one, which is somewhat reductionist, and hoping that you're going to replicate a given outcome, why not just take the gut microbiome from someone who has a low amyloid burden and transplant that into someone who has a high amyloid burden, with the hope that maybe it isn't just one bacteria, but you're getting the whole ecosystem that contributes to the favorable brain-related, pathology, right? So, but even that, there are other factors that lead to microbiome composition. And if you're missing, whether it's fiber or whatever the other variables, it could be fiber, it could be exercise, it could be age, it could be medications. That's going to then, so even though you've got a new microbiome in your microbiome, and you've got those other species that are potentially contributing to more butyrate production, if you don't have the other factors that lead to keeping those bacteria there, they're just going to go away, and you're going to have sustained growth of the variables that affect, the quote unquote bad bacteria again. So I wish it was as simple as probiotic supplementation. In animal models, that seems to work great for many things. And fecal transplantation could work too, but for me, it's getting after all of the variables that impact that to really get the best results, rather than, I see so much of science, with the probiotic and fecal transplantation approach. But again, if you're not covering all the bases, are you going to get a maximal, or any even effect? So.
And there's also the case for targeting the harmful bacteria as well. And
so that's part 3. So
yeah.
So along those lines, there's a group, Stanley Hazen, who they look at thrombosis risk, so stroke risk, and they identified. So like you were saying, they identified the bacteria that they thought were playing a role in thrombosis, and then they identified the genes that were making certain metabolites. So in this case, it's TMAO, trimethylamine oxide, which has been shown to cause atherosclerosis in mice. So anyway, they identified the genes that are produced by the gut bacteria in this process. And then they thought, all right, we're going to develop a bacterial inhibitor. And if TMAO is playing a causative role, we'll get less stroke risk, less atherosclerosis, and all of that related stuff. So the importance of that is because most of these bacterial genes that make bacterial metabolites, we don't have them. They're just exclusively bacterial. So if you take a pill and that's going to inhibit a bacterial enzyme, you're protected because the human DNA doesn't include that gene. So yeah, when they gave that specific bacterial inhibitor, and not an antibiotic, which would wipe out many bacteria, just a specific type of bacteria that makes a specific metabolite, they actually saw a decrease in thrombosis. So I actually think that's the best approach of the bunch when comparing probiotics, fecal transplants, and the targeted enzymatic inhibition approach. But to do that, you've got to know exactly the bacteria that are you've got to know exactly the metabolite, bacterial metabolite, and then you've got to know the gene that's producing that. Because you don't want to offshoot it, right? Maybe you hit one gene, but that hits 4 genes, and now you've reduced the production of good when compared to the bad. So, but then also one could argue that's too much of a reductionist approach too, because if you know that the short chains, just going back to the short chains, if you know that the short chains acidify the gut, and then because acidification of the gut limits the growth of these, bad bacteria that have these genes, well, why not just do something to boost short chain fatty acid production, which goes back to the fiber and exercise and all the things that have been shown to boost that. So
yeah, and then there's also the other thing as well. It's very often not as simple in biology as people would assume. Sometimes that there could be the case that that particular bacteria is causing a problem, but it could also be producing other metabolites that are useful, and in destroying or knocking down that particular type of bacteria, it's going to be a balancing act, because they may be beneficial in some way. To use a signaling example, TGF beta, for example, we know that at high levels it inhibits stem cell mobility and tissue regeneration, but we also know that it facilitates wound healing, right? And that's one of the concerns with things like senolytics is you don't want to do it too much, because your body needs some of these molecules at some level. So I guess it would be a case of identifying the problem bacterial populations, but controlling them, calibrating them to a level that's sort of consistent. I suppose we could use a youthful, healthy microbiome as a good sort of measuring stick, really, I suppose, and then develop therapies that tamp it down, but not necessarily eradicate those bacteria, because, as I say, they could be performing other useful
Yeah.
functions.
So you wouldn't be eradicating the bacteria. You would only be inhibiting the bacterial enzymes
Mm.
that produce these, quote unquote, bad metabolites that negatively affect our health. In this case, TMAO. But interestingly, along those lines, inhibition of the TMAO producing gut bacteria producing enzyme that produces TMAO. There are other gut bacteria metabolites that like phenol sulfate, just related to that it also inhibited. So you may get, besides it just hitting that one gene, you may actually knock out other quote unquote bad production of bad metabolites, just along those lines. So let's say you've got someone who's very old, and for them to start an exercise program is too difficult, and they don't want to change their diet. In that case, I'd argue this would be the best approach for a bacterial inhibitor to inhibit the production of some of these metabolites that negatively affect health. Just to beat that to death even more, like using that approach that Hazen's group used, these are quote unquote uremic metabolites, which negatively affect kidney health. So if your kidneys are suboptimal, they can't filter out these, the quote unquote bad metabolites that are derived from bacteria in the blood. So it's like a vicious cycle, age-related decline in kidney function, accumulation of these uremic metabolites, and then the uremic metabolites are toxic to kidney. So you've got, so the kidney function continuously declines, they continuously accumulate. And I wouldn't be surprised to see many of these gut-derived metabolites that are, like LPS, that are actually associated with Alzheimer's-related pathology. So I'd say the enzymatic inhibition approach for limiting gut bacterial metabolites that negatively affect their health, I'd say that is at the top of my list in terms of therapeutics. I expect a future where that's rampant. And actually, conversely, therapeutics that increase the expression of genes, for example, to make short-chain fatty acids, where you don't actually have to eat the fiber, you're just increasing bacterial gene expression so that they're making more short chains without the substrate. So, but that again may be a fiber thing, so.
Well, we do know that colonocytes, for example, in the gut, they rely on, for 70% of their energy, they rely on butyrate. So, if that's not a case for the permeable gut syndrome, some people call it leaky gut, although that's got a bit of should we say, a less than stellar reputation as a word, because for years people sort of refused to acknowledge the fact that leaky gut is a thing, but dysbiosis or holes or permeability, whatever you want to call it, it's a strong case for butyrate, the bacteria that produce it, and our own body cells that actually rely on it. So they use it as an energy source.
So actually, I didn't think about this before, but an alternative possibility to the gut barrier story is an alteration in blood microbiome composition in correlation with an increased amyloid burden in that study. So there is a blood microbiome. And actually, in older adults versus younger adults, there's an increase in proteobacteria in the blood. Proteobacteria are gram-negative, which means that they have LPS in their outer membrane. So independent of the gut barrier function story, which again, they didn't measure any markers of gut barrier function besides LPS, but it's possible that whatever the gut microbiome composition is, even if it's in very low amounts compared to the other sites like the mouth, which has a microbiome, or the colon, which is where most of the microbiome is, there is a gut microbiome in blood that's been shown in studies. So it's possible that the composition of that is altered. So now there's an increase in LPS containing bacteria in the blood, but still that's correlated with more amyloid burden. So identifying why that would be the case is also interesting, independent of the gut barrier function story.
It's a real rabbit hole, isn't it? So aside folks from the advice your grandma probably gave you, which is eat your veggies, don't eat too many candies or sweets, as we call them over here in the UK, and get plenty of sleep, I'm not sure what we can we can do really. I would just try and eat more fiber in general, stay active, right?
Exercise, yeah, that too. That should help.
And hopefully the researchers will home in on the culprit, and people like Mike as well who are doing similar related work. Well, and in the next few years, hopefully we'll have a clearer picture of what's actually going on, and more importantly, be able to develop sort of interventions to prevent things like Alzheimer's, or certainly bacterial driven Alzheimer's. So fascinating paper, fascinating discussion as always, Mike.
Yes, Steve.
We could probably do a regular show with the amount of stuff that we both discussed. But thanks for coming on today and sharing your thoughts. I thought it was an exciting paper, and that's my personal view. I'm allowed to think things are exciting, and obviously the audience at home. Hopefully you found it interesting, if nothing else. And thanks very much for joining us, Mike.
Sounds good. Thanks, Steve.
I'm sure we'll probably see you again in the near future.
Definitely. All right. Take care. Talk to you soon. All right. Ciao.